This protocol outlines the production of immunotherapeutic rhesus T cells that express two proteins, an antiviral chimeric antigen receptor and a chemokine receptor, CXCR5, that target cells to lymphoid follicles. This relatively rapid 9-day procedure produces T cells with a less differentiated central memory phenotype with the potential for long-term persistence after infusion into animals.
Adapting this method to human cells will allow production of immunotherapeutic T cells with the potential to induce a durable remission to HIV without administration of antiretroviral drugs. This method can be used broadly to produce rhesus T cells expressing other proteins of interest, and with minor modifications, it can be used to generate transduced T cells in other species, including humans. The success of the transduction is dependent upon healthy, stimulated PBMCs. Care must be taken in collection, transport, and storage. Prior to transduction, it's important to visually assess stimulation of the cells.
Start by thawing primary rhesus PBMCs. Incubate the cells in a 37 degrees Celsius water bath with gentle agitation until only a small amount of ice remains. Gently pipette the cells into a conical tube. Then rinse the vial with 1 milliliter of basic medium and slowly add it to the cells. Slowly add an additional 9 milliliters of warm basic medium to the cells. Place the tube in the aerosol-resistant canister and centrifuge it at 600 times G for 5 minutes at 22 degrees Celsius. Aspirate the supernatant and resuspend the pellet in a small amount of growth medium.